So since we've already gone through mitosis, and I know you've studied that really, really hard, no. Since it's going to be on the final, you're going to begin to do that. But as you study mitosis, especially over the weekend, and go ahead and look ahead at those chapters on transcription, translation this weekend and get ahead. We're going to talk now about meiosis. And the illustration here on the front really brings home what we're talking about. Because at the point of a fertilized egg, you have a full complement of chromosomes. And how many chromosomes does a human cell have in it? 46 chromosomes. You have 23 pair of chromosomes. You got 23 from mom, 23 from dad. So since you have 2 complete sets of your genome, we say that is 2N or diploid. So all of our cells are going to be diploid. As we go through mitosis, when a parent cell divides into 2 cells, how many human cells, daughter cells, how many chromosomes are going to be in each one of those daughter cells after mitosis? 46. 23 pair. Because mitosis is what? What's the word I put with mitosis? Well, it's diploid. It's moving from a diploid cell to 2 diploid cells. But if you make an exact copy, cloning. That's the word. Making an exact copy. It's cloning. But you also understand that when we have a sperm and an egg, we have to halve the genetic complement. Because these 2 cells are going to come together to form a unique individual. So the process of meiosis is how we take a diploid precursor cell, an oogonia in females and a spermatogonia in males, and we end up with gametes, sperm and an egg, that only have 23 chromosomes. They have 1 set. So when they come together, that gives you the final 2 sets for the individual. And so with just 1 set of chromosomes, we say that's haploid, or 1N, 1 copy, N being the number of chromosomes. And so we're looking at this meiotic division not being cloning, but being a reduction division. We're cutting our number of chromosomes in half. But the other important thing to remember about meiosis is the chromosomes that an offspring is getting from dad and the chromosomes that the offspring gets from mom aren't exactly the same. Because there's going to be some recombination, some exchange of genetic material. So meiosis is about a reduction division and recombination that leads to a unique individual that never existed before in the whole evolution of life on Earth. So that's where we're going today. So let's get some vocabulary down as we begin to look at meiosis. Are we good with what a gene is? Somebody tell me a gene that we have in the human body, human genome. Tell me a gene. Eye color, hair color, skin color. Yeah. Now, we're going to get to that term in just a bit. But we have genes that encode for eye color, hair color, skin color. Maybe some genes we don't like that predispose us to certain diseases. So a gene is a specific segment of your DNA that encodes for a trait. And we're going to distinguish between the gene and the trait and what it looks like based on what you said. Locust sounds like location. It's not that bug that grows on the side of the tree here in East Texas, so we run into trouble with that word. Locus is location. Where is this particular gene on a chromosome? Now, somebody said eye color. Who said eye color? I heard somebody say eye color. How many different, oh, I said it? Yeah. Okay, well, I'm not going to answer my own question. How many different genes do we have for eye color? Give me an example. Brown, blue, green. Okay, so all these variations of one particular gene is referred to as an allele. And so when you look at your genome, you're going to have 2 alleles for every gene. You get 1 set of chromosomes, therefore you get 1 allele from dad. You're going to get a different allele from mom. So in our picture, we have some chromosomes, and we have certain genes on those chromosomes, and these genes can either be for blue eyes that you see with the blue stripe, or brown eyes with the brown stripe. And you can see those alleles may be different. They may be the same for brown, or they may be the same for blue. But that's still the gene for eye color, and where it is on the chromosome is the locus of that particular gene. So are we good with those 3? The distinction between those 3 terms? Dominant. This is an allele that when it is present, it doesn't matter what the other allele in your pair may be. That's the gene that's going to be expressed and result in a physical manifestation of a trait. Whereas a recessive allele, that's only going to be physically expressed if both alleles are recessive. There's not anything drowning it out. So think of a dominant allele as being the extrovert and the recessive alleles being the introvert. And the only way an introvert's ever going to talk is if there are 2 introverts together. I don't think 2 introverts never say anything to each other, but does that analogy work? Now, our practical example of this is earlobes. So when we look at detached earlobes, which are the earlobes that just hang down, and you got this little part here, okay? That is a dominant gene, a dominant allele. And so this detached earlobe, that physical manifestation of it, means you have at least 1, or you may have 2 dominant alleles, but all you need is 1. But the attached earlobe that comes straight in and you don't have this little droopy part, the attached earlobe, you have 2 recessive alleles, and that's the only way you get that expression. You see how my earlobe comes down, droops down, and then it goes back up and attaches? Okay, that's a detached earlobe. People have the earlobe that it just comes down and it just goes straight in to the side of your head. All right? Most people are going to have the detached, but there are going to be some that will have the attached. Now, we do have other genes that can be expressed together. Now, if you printed out the PowerPoints, I've changed them up a little bit, and I'll get the new PowerPoint loaded up this afternoon. But an example of what we say is codominant is when each allele is expressed regardless of what the other allele may be. And so the example I like to use is our ABO blood types. And so when you look at ABO blood type, if you have type A blood is one of the more common blood types you have. You're either going to have 2 A alleles, or you're going to have 1 A allele and an O-type allele. Now, when you get to physiology, you're going to learn O-type allele does not mean there's nothing there. There's actually a protein called the H antigen. The H antigen is the base upon which all of your other types are going to form. The difference between O-type blood and A-type blood is 1 sugar added. That's it, 1 additional sugar. The difference between A-type blood and B-type blood depends on the type of sugar that is added to the H antigen. So for a person that has an A allele and an O allele, they've got the A antigen and the O-type antigen expressed on their red blood cells, both together at the same time. But the only one that your body can recognize to possibly make antibodies against is the A antigen. You're not going to make antibodies against O. But that's physiology. B-type blood is either 2 Bs or a BO allele. But here's where you really see the codominance. Anybody have AB blood? That's not a hugely common type. You have AB blood because you have 1 allele for A and 1 for B. You got 1 from mom, 1 from dad. But you express both on your red blood cells together. That's the codominance. And then O-type blood, you have to have 2 O alleles so that you don't have the A or the B.
Is it still codominant if mom is AB and dad is B, and then child comes out AB, or is that just
No, if your mom is AB and dad is B, then you got the A allele from mom and the B allele from dad.
Oh, still?
Yep. Because you can get either, it's a 50-50 shot what you're going to get from mom and a 50-50 shot from dad.
Well, why would it be AA if they both have A?
No, mom has AB, dad only has B.
Oh, okay.
And the B for dad is either 2 B's or a B and an O. So with that setup, you could have a child born with A-type blood, a child born with B-type blood, or a child born with AB blood. Okay? Because you're going to get an A or a B from mom, and you're going to get an O, which would allow either one of those to be expressed, or a B from dad. Okay? Now, in our slide where we saw the chromosomes, if you have the alleles the same on both set of your alleles, we say that's homozygous. If the alleles are different, like an AO or a BO, that's heterozygous. Homo, same, hetero, different. And again, we're still looking at those 2 alleles, and here we're still looking at our gene for ABO blood type. Now, here's what you had suggested earlier when we were talking about gene for eye color, hair color, skin color. The actual DNA sequence that encodes for those traits is called the genotype. But when you actually see the physical manifestation of those genes, so blue eyes, brown hair, dark skin, then that's the phenotype. And in our chart, again, of blood types, here we have homozygous A, heterozygous A. These are all your genotypes you could have from our parents. But when we look at your blood type, these are the phenotypes. A-type blood, B-type blood, A, B, and O. Those are your phenotypes. And you can have 2 different genotypes for A, either an AO or an AA. You can have 2 different genotypes for B, either a homozygous B or heterozygous BO, but you can only have 1 genotype that will yield the phenotype of O-type blood. Do you see the distinction? Genotype, gene, phenotype, physical. Okay? The G and the pH. That's how you can keep those aligned in your mind. Now, so far, when we talked about mitosis, the cloning of 1 cell into 2, we were primarily talking about somatic cells. And when we talked about neurons, what was another name for the soma of a neuron? The body. Soma means body. So when you say somatic cell, what kind of cells are we talking about? Your body cells, the majority of the cells in your body. And we're going to distinguish those cells that are undergoing mitosis from our germ cells, which are the cells that are going to yield a sperm or an egg. So we're talking about very, very few cells in the human body. These are the spermatogonia and the oogonia that are undergoing meiosis to yield a sperm and an egg. And so here is our mitosis, 1 parent, 1 set of divisions. And what are our stages for mitosis? What's our 4 letters? PMAT. You haven't even studied and you know that. PMAT. You do PMAT 1 time and you end up dividing that parent cell in half, 2 exact copies of the parent. For meiosis, we're going to do PMAT twice. We're going to do 2 divisions. You go through PMAT 1 and you end up with 2 cells that are sort of intermediates. You go through PMAT 2 and you end up with 4 daughter cells that result, each of which has half of the number of chromosomes that we got from the parent. So essentially 1 copy of every chromosome at the end of meiosis, and the parent cell that you start with had 2 copies of each chromosome.
So you're still 2 copies at the first cell division?
Yep, we're 2 copies right here. And here, 2 copies-ish. We're going to have to modify that. And we're going to step through it so it's very clear what you got here before you do it again. All right? Diploid, haploid. We've already talked about that from our introductory side. Diploid, 2 copies of every chromosome in the genome. Haploid, 1 copy of every chromosome in the genome. Now, I want to go ahead and introduce this concept that we're going to keep throughout our talk. Because in mitosis, what lined up right along the equator during metaphase? What did we line up? All the sister chromatids. And we've got our pairs of sister chromatids. How many pairs of sister chromatids lined up right along the midline? 46. And we pulled those pairs apart into 46 chromosomes going one direction, 46 chromosomes going another direction. In which phase? Anaphase. That's the A in PMAT. Now, in meiosis, we're going to be moving these pairs of sister chromatids around. And one organization, one level of organization we need to be familiar with are what are called homologous chromosomes. What does homo mean? The same. So homologous chromosomes, are you already thinking if they're homologous, they must be the same? So homologous chromosomes would be, if we look at this pair, we're going to say a homologous chromosome is chromosome number 1. How many chromosome number 1s do you have in all your cells? Remember, you're diploid. So your chromosome number 1, you have 2 copies of it. You got 1 chromosome 1 from mom and 1 chromosome 1 from dad. And when you do your karyotyping, that's why they're situated out in pairs, those 23 pairs. So chromosome number 1, 1 from mom, 1 from dad. I think you got it backwards. That is a homologous chromosome pair. And so in one division of meiosis, we're going to separate homologous chromosomes into opposite daughter cells. That's going to be our first division. But we've already replicated them, so they're actually going to exist as sister chromatids. There's going to be a sister chromatid pair from mom for chromosome 1, a sister chromatid pair of chromosome 1 from dad. And we're not pulling those sister chromatids apart in the first division of meiosis. We're simply moving one of those homologous chromosomes that exist as sister chromatids and the other one. We're moving them away, separating. And then in the second division, the second PMAT of meiosis, it's going to be almost exactly like mitosis, but we're just working with half the number of chromatids. We're going to pull those apart. So in PMAT 2, it's similar to mitosis, only in the sense the way it's different. In mitosis, we separate 46 sister chromatid pairs. In PMAT 2, we're only separating 23 because we divided them in half in that first division. Okay? That's a preview of where we're going. We're going to walk through all that. But it's important to understand that our homologous chromosomes have already gone through the S phase of interphase. They have replicated themselves. So as we're looking at meiosis, we're going to be calling this thing a homologous chromosome of each other. But what do they exist as? Sister chromatids. Okay? Does everyone see that? All right. That's going to be important for understanding where we're going and how we're going to get there. So as we look at preparing cells for reproduction, we can have this asexual reproduction. And asexual reproduction in some, unicellular organisms, it's going to be asexual. You simply pinch it off, binary fission in bacteria. I mean, that's much like mitosis. That's cloning. So your unicellular organisms, you can bud, send off little parts and pieces from multicellular organisms. That's all mitosis. We're not talking anything like that in our eukaryotic cells. But we're talking about this reduction division, this 2 rounds of PMAT. And again, remember interphase? We're going to have our 3, what are our 3 stages of interphase? G1, S, G2. We're still doing that. But when we get to that M for our germ cells, that's meiosis, not mitosis. Okay? So we're going to duplicate that DNA. We're going to form those sister chromatids. But do you see how even in this illustration they're calling them homologous pair of chromosomes? It's easier to say chromosome than always having to say homologous pair of sister chromatids. But when you see that X, you know those are chromatids joined at the centromere. Okay? So, meiosis, 2 PMATs, 4 haploid cells. Now, I'm going to introduce this concept that we're going to hammer home again as well. If we're doing 2 divisions, when you think about the cell cycle for mitosis, all right, G1, S, what happens in S? DNA synthesis. G2, mitosis. If we wanted to do mitosis again, what would we have to do? G1, S, what is synthesized? DNA, G2, and mitosis. That's how we do the cloning. Between the first PMAT and the second PMAT in meiosis, listen carefully, write this down. Between PMAT 1 and PMAT 2 in meiosis, there is no DNA synthesis. Between PMAT 1 and PMAT 2, there is no new DNA synthesis. That's how you cut it in half. If you had another synthesis stage there, it would just be the cloning thing again with some, a little bit of reorganization. That's another of the keys to meiosis. There's not a new synthesis of DNA between the first and second divisions. Correct. That's how we end up with haploid cells instead of diploid daughter cells. So we're going to start with meiosis 1. Again, I like to call it PMAT 1 because guess what? Same stages. Prophase, metaphase, anaphase, telophase. It's going to look pretty much the same, but we're moving things around a little bit differently. We're also in prophase 1. That can be divided into 5 stages: leptotene, zygotene, pachytene, diplotene, diakinesis. Kinesis being movement. And the movement that's happening here is our spindle formation. The nucleus is breaking down. But the one substage of prophase 1 I want you to know is pachytene. Pachytene, this is the one that you absolutely have to know. The others are important. This one is infinitely most important in my mind. Because in prophase 1, something happens in prophase 1 of meiosis that doesn't happen in mitosis, and it doesn't happen in prophase 2. You have a mechanism that is called crossing over. That's where the arms of your homologous chromosomes get twisted together and they can exchange materials. So that mom's chromosome can actually trade a little bit of its DNA with one of dad's homologous chromosomes. And that way an offspring is going to get a chromosome that's partly mom and partly dad. And so it's not like either one, but like both. And it's unique for that individual. That crossing over event happens in the pachytene substage of prophase 1. Everything else about prophase is the same as what you see in mitosis, except for crossing over that's happening in the pachytene substage of prophase 1. So you learn prophase and mitosis. You just say, okay, prophase 1, the same. We're just adding that part. We'll talk about crossing over in more detail Maybe we can get to it today, I'm not sure. But again, when you look at what's happening in prophase 1, nucleus is breaking down, we're condensing our chromosomes, we're starting to form the spindle apparatus. What does that all sound like? That just sounds like prophase and mitosis. But it's that Pachytene stage where we get the crossing over event. Do you see how the blue and the red have exchanged a little bit? That's the significance. That's the recombination that we're talking about. That really pumps up the variability of the human species. So, we take our homologous chromosomes and we line the pair of homologous chromosomes up on either side of the equator of the cell. But all of our homologous chromosomes are paired and aligned along the equator of the cell. And when you see chromosomes condensed and at the middle, what do we call it? Metaphase. In this case, your pair of homologous chromosomes, that's metaphase 1. And do you see how they're stacked up on top of each other? One north, one south. Mitosis, they were all lined up in a straight line east to west. But when you stack the pairs up and you see those pairs exist as sister chromatids, you know you're in metaphase 1. We have our spindle fibers. We've got the kinetical. Everything is the same as we see in metaphase of mitosis, except we have our 23 pair of homologs lined up on either side of the equator instead of all 46 lined up in a straight line. All right, so we got the alignment. We see the difference. Mitosis, 46 sister chromatids in metaphase for mitosis. In meiosis 1, we've got our homologous pair of sister chromatids on either side of the equator. What comes after metaphase? Anaphase. So when we look at anaphase, homologous chromosomes are moved. You may see some books that say separated. I don't like that word because we're not pulling the sister chromatids apart. We're moving our homologous pair. You see they still exist as sister chromatids. We're moving this chromosome 1 to this cell. This chromosome 1 is going to the other cell. But it still has its copy with it, so they're still sister chromatids. But they're no longer at the midline. They're moving apart from each other. Can't say that word. So that's anaphase. What comes next? Telophase. Our sister chromatids arrive at each pole. Now, how many pair of sister chromatids do we have north and how many do we have south? 23. 23. Do you see how we've already cut it in half? So the reduction happens in PMAT 1. The crossing over happens in Pachytene of prophase 1. The reduction happens in PMAT 1. PMAT 2 is going to look just like mitosis. We just have half the number of sister chromatids to pull apart. That's it. Bless you. So there's our meiosis 1 summarized. So you see, and I like the fact that they don't use that pro-meta term like they did in mitosis. They've got early prophase, late prophase. That's why I didn't want you to even fool with that with mitosis. It's prophase until they line up at the middle, which is meta, moving apart, ana, starting to reform and uncoil, that's telophase. And we have our cytokinesis event. So we end up with 2 daughter cells. Now, how many, in fact, chromosomes do we have in each one of our daughter cells? Remember, there are 23 pair of chromatids. So, in fact, if you could separate those sister chromatids, how many chromosomes we got? 46. I think that's what you said. 46. But how many different chromosomes do we have? Only 23. And so since there are only 23 different ones, even though those 23 have a copy, so they're officially 46 chromosomes if you could separate them, A lot of people call those haploid cells at the end of the first division, even though there's still 46 chromosomes in there. But you only have 23 that are different. They just have their copy. So it's okay to call them haploid, because you only have one copy of the code, even though you've got two sets of each. Make sense? That's a little kind of hard to grab a hold of. Meiosis 2, this is a piece of cake. Looks a lot like mitosis. You're going to split those cells in half. You're going to separate the sister chromatids now. All 23 of those sister chromatids are going to line up at the equator at metaphase, and they're going to get pulled apart. But that's the key to the maintaining that half number, that haploid number. No DNA replication between PMAT1 and PMAT2.
So prophase 2 is the exact same.
Yeah. Prophase 2 is going to look exactly like prophase of mitosis. The only difference, well, that's how it differs from prophase 1 of meiosis. But prophase 2 of meiosis and prophase of mitosis is going to look the same, except in mitosis you're dealing with 46 pair of chromatids. Prophase 2, you're only dealing with 23 pair of sister chromatids. So here we go. Prophase 2, breaking down the nuclear envelope, forming our centrosomes, centrioles, organizing the microtubules into spindles, attaching to the kinetochore, moving our sister chromatids around. Metaphase 2, line up right along the midline. Now notice, these 23 are lined up side by side all along the length, just like in mitosis, but the number is 23 instead of 46. What's after metaphase? Anaphase. The sister chromatids are pulled apart. How many sister chromatids did we have? 23 in this one, 23 in the PMAT2. When you pull the sister chromatids apart, how many is this daughter cell going to get? 23. How many is this daughter cell going to get? 23. That's the end of the process in which each daughter cell ends up with 23 chromosomes. Telophase, just like telophase in mitosis, we're going to start to uncoil the DNA. Nuclear envelope's going to start to form. Microtubules break down. We start to undergo cytokinesis. And that ends meiosis. Easy. Once you've got PMAT, PMAT1, PMAT2, understanding the distinction between homologous chromosomes in PMAT1 and the sister chromatids in PMAT2, just like sister chromatids in mitosis, it's a piece of cake. I mean, it's a piece of cake. Now, this whole ordeal is to make a sperm and an egg, right? So we call the process of forming your gametes, a sperm and an egg, gametogenesis. You see where we get the name? Gamete, genesis, the formation of. So you undergo meiosis. And at the end of meiosis, starting with a spermatogonia, you end up with 4 spermatozoa that have half the number of DNA than the cell that they started with. Think of this as a stem cell. That's spermatogenesis. Now, ladies, you do something different from guys. Have you noticed guys and girls are different? What is that? Men are from Mars, women are from Venus? Okay. Here's another distinction between male and female. During oogenesis, which is the process of forming an oocyte or an egg, at the end of meiosis, you do have 4 nuclear daughter cells. But all the cytoplasm of the egg stays with one of the daughters. The 3 other nuclei with just a little bit of plasma membrane around them exist as polar bodies. So you have 4 haploid products of oogenesis. It's just that the cell stuff, the cytoplasm, all the organelles stays with one of those daughters, and that's going to be the egg. Because how many eggs do you need to get a baby? 1. How many sperm do you need to get a baby? You need a bunch. You need a whole lot. Because guys, is this you and me having to take the heat here? Okay, I'm sorry. I'm sorry. Yeah, I was panicking because I'm like, where's Mr. Blue over there? And okay. First of all, here's and the reason you need a bunch. The first thing that's going to happen when you ladies ovulate, you ovulate from one ovary every other month, typically. But both ovaries are not going to ovulate at the same time. So, the first thing that happens when there's an ovulation and after you have had intercourse and sperm get introduced in the female reproductive tract, where do half of the sperm go? The wrong way. Guys don't ask directions, right? No, we don't ask. That's weak. So the very first thing that happens is the sperm get divided in half. So you have half as many. There are going to be a lot of sperm that don't have enough energy to get to the end of the process because fertilization occurs at the very distant end of the fallopian tube adjacent to the ovary. It's called the ampulla of the fallopian tube. Fertilization must occur at the end because it takes that fertilized egg several days to make its way into the uterus. And over the course of those days, those cells are dividing and it produces a structure that's going to help that fertilized, it's called a zygote, that zygote attach to and dig into the endometrial lining of the mother. If fertilization happens in the middle of the fallopian tube or in the uterus itself, it's just going to wash out of the female reproductive system and you won't have a successful pregnancy. So, sperm get divided in half. A lot of the sperm don't have the energy to get to the end. There are a lot of lazy sperm. Ladies, don't you say one word. There are a lot of lazy sperm. But you have this whole shell around the oocyte. You have this protein shell called the zona pellucida. You have a bunch of cells surrounding it called the corona radiata. So it takes a lot of sperm coming in with all these enzymes to degrade through the cells to drill a hole through the zona pellucida. So you got a lot of these kamikaze sperm that are hitting and they give off their enzymes, but it's not enough to get all the way through. So survival of the human species is left up to that male sperm that stopped off at Starbucks and got a coffee and read the newspaper and let all these other sperm dig the hole, and then he comes in at the last minute and goes, okay, let me get in here, and he fertilizes the egg. Does that give you some real good confidence in males and survival of the species? It's the lazy late sperm that makes the new individual. But you need a lot of sperm, but you only need one egg. But only one sperm is actually going to fertilize and create a new individual. So when we look at the eggs, that process is going to start when you're born, ladies. You're going to form all of those oocytes, 500,000. But most of those eggs are going to be paused. And they're going to arrest in the middle of that cell cycle, and it may last your entire life. Because you're not going to mature every egg that you're born with. What you're born with, you're not going to make more. So your eggs are held in that first meiotic division, PMAT1, and then there's a pause until there's a hormonal signal to go ahead and do the second meiotic division.